hey, let's draw a skeletal muscle cell. In fact, this section of our lecture, hey, let's draw a skeletal muscle cell, I'm hopeful will help us out in the understanding of the nature of these cells. So what I would like you to do, fellow A&P nerds, is get yourselves ready, get a blank sheet of paper, maybe orient it landscape orientation so you have more room to draw. Get yourself some colored markers, colored pens, various colors if you have those. Pause me if you need to get ready, and then we will proceed to draw a skeletal muscle cell with me blabbing on about it somewhat because I'm a professor. We blab. That's what we do. And hopefully this will help you some, at least, in the understanding of the physiology of these skeletal muscle cells. So again, hey, let's draw a skeletal muscle cell. So here I have a blank canvas, let's call it. Mine happens to be digital. Yours is probably an actual piece of paper. And what I would like you to do first, everyone, because trust me, I know at least this much about your personality. You would hate it if I come back at the end and add something onto a picture that we didn't leave room for ahead of time. So what I would like you to do is on your diagram, leave maybe, if I was judging, the top quarter of your page blank. So watch where I draw this dashed line. Don't draw this line, but leave the top space here empty because we're going to put some stuff in here later. So when we construct our muscle cell, we'll start drawing it a little bit lower, so about 25% down your page is what I recommend. And I'm going to start with red, a color red for my picture on my canvas. And I'm going to start with a somewhat straight line going across my page. And then about right here, I'm going to stop. I'm going to put a little circle here. And I'm going to continue on with my picture out here somewhere. Great, that's beautiful. From my little circle, I'm going to draw two vertical lines down. And then another circle. Like that. And then some straight lines. I bet you know what I'm drawing here. I bet you can figure it out if you've been studying your A&P. Have at the ready any set of steps you have in the contraction of a skeletal muscle cell and a good diagram from your textbook of a skeletal muscle fiber, like this one. And remember, when we use the word, I'm going to write this and then erase it, fiber, that's the same as using the word cell in the muscle biz. So one muscle fiber is the same thing as one muscle cell. And what I've drawn here for us is, of course, I bet you knew, the sarcolemma. The cell membrane of a muscle cell. The sarcolemma, like that. That's what all this red stuff is. And right in the center, right here, see, I bet you figured this out as you've been doing your studying. Right here, I have a T-Tubule, or transverse tubule. That inward projection or invagination of the cell membrane that runs straight through the entire width of this cell perpendicular to the long axis. That's what these T-tubules do. So far I've got sarcolemma and T-tubules. And what is it that we have right next to the T-tubules? Butted right up against the T-tubule. I'm going to draw it right here. I'm using orange here just so it stands out a little bit. And I'll go like this right here, and then another one living right here on the other side. Like this. Now, as I do this, people, understand there isn't an easy way, a trick to any of this. I'm suggesting you actually make a picture like this. Oh, it's great. You can watch me draw one or watch any old professor draw one, but it's much better if you make one yourself, label it to look at, maybe even make it 3 or 4 times so that you have one to study with. The creation of these drawings is what helps you understand. This is my point. What I've drawn right here is the SR, the sarcoplasmic reticulum. You might remember that specialized type of endoplasmic reticulum that muscle cells have. And what does sarcoplasmic reticulum do? It stores and releases calcium ions. You've probably already covered this in your lectures. Stores and releases calcium ions. In fact, technically, wouldn't the 2 pieces of sarcoplasmic reticulum I have here and here that butt right up against the T-tubule, wouldn't these technically be called terminal cisternae? I'm going to say that plural. Terminal cisterna is one, terminal cisternae, more than one, and it would continue all the way throughout the cell. So there'd be sarcoplasmic reticulum all over out here, people. I'm not saying that this picture I'm making for you is histologically accurate. This is a stylized picture of a skeletal muscle fiber that we use that hopefully helps you understand the functioning of the cell. So, my triad, terminal cisterna on the left, T-tubule, terminal cisterna on the right. And inside here, I might as well, inside the sarcoplasmic reticulum, let's put in our calcium ions. I'll just stick with my orange color. Calcium ions. Bunch of calcium ions in here. Because that's one of the jobs of sarcoplasmic reticulum, is to store up calcium ions. So this is where all the calciums are. Inside the sarcoplasmic reticulum. Now, off to the right, in this blank spot that I have, I'm going to draw a great big, ginormous, giant, massively big sarcomere. Remember what I said, this is a stylized muscle fiber, not cellularly accurate. But I want you to think about, what is it I'm drawing here, everyone? What the heck are these things? Well, I think I know. On either side, I have my Z discs or Z lines, depending on the textbook you look at, and my actin proteins right here as these horizontal black lines. So I've drawn in a bunch of actins here, 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 and Z lines. Now, understand this sarcomere is way, way, way too big to be realistic, isn't it? Look at any picture you have in your textbook. Wouldn't you, and please do not draw this part, I'm just going to draw it and erase it. Wouldn't you know that inside this cell we would have a bunch of myofibrils like this, each one containing many sarcomeres like that, you know, like this, so on and so on. So what I've done is drawn just a great big giant one for us to look at. It's not realistic size-wise, but I think you get the point. And are the actin proteins the only part of these thin myofilaments? Well, no, we know better than that. Don't we have those tropomyosin ropes that go around them? See why I told you to get different colors? Use a lot of different colors if you can, because it just makes it easier for you to study with this picture. These are the tropomyosin ropes covering up the active sites on the actin filament. So I'm just going to label this for myself. Tropomyosin, right here. And is that all? Well, no, I think we know better than that too. Because what else do we have protein-wise in the thin myofilaments? We have these troponins, proteins that are attached to the tropomyosin rope every so often. Hopefully you covered these in your lecture. Look at the pictures you have in your textbook. But I suggest you draw them in sort of like this. My picture, they look sort of like pink jelly beans or something, don't they? And what are those? Troponins. Or troponin, you can say it that way too. Remember that the root on these two words, tropomyosin and troponin, refers to the fact that these are moving proteins. Still not done with my sarcomere, however, because what else do I have in here? Well, I would have to have the thick myofilaments, like so. Like so. I'll shade these in. Why not? And these are the proteins that have the heads on them. These are the proteins with the heads. What are these? Myosins, of course. I don't know if mine are that pretty, but I think you get the point. And what are those green ones? Myosins, the thick myofilament. So that's myosin right there. And what is it, those myosin heads, if you want to give, a section of a protein myofilament a personality, what is it those myosin heads want to do? They want to grab onto the active sites on the actin filaments and yank them. They want to grab those things and pull them in this direction toward the center of the sarcomere, don't they? They're just not able to most times because why? The active sites are covered by what? The tropomyosin ropes. This hopefully is not news to any of you. If it is news, you need to study your steps a little bit first before you keep working on this picture. So I've got a sarcomere there. Oh, and don't forget, of course, I would have some titin proteins in there. I'm not going to draw them, but they'd be, well, I'll just draw you one. So they'd be sitting in here, right, anchoring everybody, giving the elastic nature to this tissue. So here we have a giant sarcomere inside a muscle cell with a sarcolemma and T-tubules and sarcoplasmic reticulum with calcium inside. And the idea is we need to get the calcium to move from here over to there, don't we? We need the calcium to go from inside the sarcoplasmic reticulum to the troponins. Because when calcium binds to troponin, it makes it pivot, roll over, fall down, however you want to think of it. Again, I'm hoping this is something that you covered in your lecture. Let me give you a quick little mental image, perhaps, that might help us out here a little bit. So here I am at my desk, and let's say that this is a pen case for my fancy tablet that I'm drawing with, but let's say this is a troponin protein or troponin protein. Again, you can say it either way, that's fine. And let's say that this little pink marker cap is a calcium ion. These two substances, the troponin protein and the calcium ion, have a degree of chemical affinity for one another. So if there's a calcium floating by like this through the cytoplasm or sarcoplasm of a muscle cell, it'll do this. It'll stick to the troponin protein. And by doing that, it causes the troponin to change its shape. And the troponin will fall down, pivot, rotate, reorient. There's lots of verbs people use for this. But here comes a calcium, if it's loose, binds to troponin and makes it fall over. Since the troponins are scattered along the tropomyosin ropes, if I could arrange to have all the troponins fall over at the same time, that would heave the tropomyosin ropes off of the active sites, allowing the myosin heads to grab and pull, which is what they want to do all day long. So I have to get these calciums loose, floating around inside my muscle cell. I have to somehow get the calciums released from the sarcoplasmic reticulum. Back in our wonderful, colorful picture, so I got to get those calciums somehow liberated from this sarcoplasmic reticulum. And how would I get an ion to move through a membrane, you might ask? Well, I would use an ion channel for that. This is one of the reasons we left the top part of our picture blank, everyone. This is one of the reasons we did that. So, let's see, what would be a good color here? How about lava? I'm going to try lava. I don't know if I've ever used that. So up here I'm going to color code. Ooh, look at that. That's kind of cool looking, isn't it? Lava. Lava will represent for me voltage gated calcium channels. Voltage gated calcium ion channels. And let me show you where they live. Right here, right here, right here, all along the sarcoplasmic reticulum. These are in lava voltage gated calcium ion channels. I'll throw some out here too so you can see them. They're all over my sarcoplasmic reticulum. Voltage gated calcium ion channels. This is how the calcium gets out. The calcium ions would move right out through these doors, loose into the cell's cytoplasm. So here's my question for you fine people. What would make a voltage-gated calcium ion channel open? Some sort of voltage change, some sort of electrochemical event, wouldn't it? So once I can make them open, the calcium will leave, go out here, bind to troponins. See, there's a troponin with a little calcium on it. And if I could do that on every one of them, then they would all flop over, exposing the active sites by pulling the tropomyosin ropes off the active sites, allowing the myosin heads to grab and pull, grab and pull. So the problem I have to contend with is opening up these voltage-gated calcium ion channels. What makes them open up? An electrical event. An electrical event where? In the T-tubules. They butt right up against the terminal cisternae, the sarcoplasmic reticulum, don't they? Which means, in my picture, I need another ion channel that I'll make dark blue. Why not? So here's another ion channel. I'll color code dark blue. If you don't actually have colors at home for making this picture, I would suggest that you use different symbols. Maybe voltage-gated calcium ion channels are X's, and these next ones are O's, like that. Here, I'm going to have voltage-gated sodium ion channels. Voltage-gated sodium ion channels are in this blue color. And I would like you to watch where I put them, everyone. Watch where I put them. I'm going to start over here on your left, maybe about right here. And I'm going to start putting in voltage-gated sodium ion channels. And look where I put them, all over the sarcolemma. Both sides. Voltage-gated sodium ion channels. And down the T-tubules, at least a little bit. I'll put some on either end here. Believe it or not, there's actually some debate on how far down T-tubules in various muscle tissues these voltage-gated sodium ion channels go, but they go down there a little bit. They go down there a little bit. In fact, I'm going to draw a little zoom in of this in just a few minutes to show you in a little more detail. So I've got voltage-gated calcium ion channels and voltage-gated sodium ion channels drawn in my picture. Now I know I sound like a broken record. That's okay. It's part of lecture to help us understand. But if we look at these calcium ions, remember, I got to get them out of the sarcoplasmic reticulum and over to my troponins. I must open these voltage-gated calcium ion channels. To make them open, I need a voltage wave. What voltage wave? The one that's going to run down the T-tubule, like that. That voltage wave originates through these voltage-gated sodium ion channels. And what would make them open? Again, a voltage wave, a change, an electrochemical event. So if I can make the voltage-gated sodium channels open, then the voltage-gated calcium channels will open. Then the calciums will flow freely into the cytoplasm or sarcoplasm of the cell, binding to the troponins, causing the troponins to pivot, roll over, fall down, pulling the tropomyosin ropes off the active sites, allowing the myosin heads to grab and pull, grab and pull. I'm just repeating the same thing over and over again. This is what we call studying, everybody. So, how do I get those voltage-gated sodium ion channels open? Well, I need another ion channel. And I think I'll use for this, how about yellow, just so it stands out. So I'm going to color code my key again in yellow. And in yellow, I have ligand-gated sodium channels. You could also say ligand-gated, you could also say chemically gated, or substance-gated sodium ion channels. I'm going to make those this weird yellow color, and I want you to look where I put them. Right here, right here, right here, and here, like this. Only over in this general area in the upper left. Ligand-gated sodium ion channels. These are new to us, right? They're not voltage gated. An electrochemical event does not cause these things to open. What would cause the opening of a ligand gated ion channel? Well, again, I'm hoping this is not news to you, but if it is, that would be a ligand. It would be a substance. Some substance has to do this. That substance we call a ligand. And I bet many of you know what ligand it is. I'll pause while you think about it or look it up. In fact, just hit the pause button because it doesn't bother me at all. I am not offended in the least if you pause me, so go ahead and pause me. Hopefully you looked it up and found that it's what? Acetylcholine. Acetylcholine is the ligand that will cause the opening of these ligand-gated sodium channels. Where does the acetylcholine come from? This is why we left the top quarter blank. How about gray? So watch what I'm going to draw in here, everybody. Right here I have the end plate of a motor neuron. A nerve cell that controls a muscle fiber is called a motor neuron. And this motor neuron right here spits out good old trusty acetylcholine. ACH. Spit out from this neuron directly onto those ligand-gated sodium ion channels like so. Causing them to do what? Open. Oh, but wait, everybody. Hang on just a minute, because your good old professor left something out, didn't he? On purpose. I left this out. I'm talking about these ion channels, voltage-gated calcium, voltage-gated sodium, and ligand-gated sodium. We know where the calcium is. See that? It's in the sarcoplasmic reticulum. But where are all these sodiums? Where are they? This is my snapping fingers. Who's working on my cell membrane? Establishing a resting membrane potential. Pumping 3 sodiums out and 2 potassiums in over and over and over again. Yes, the sodium-potassium pump, everybody. Before you're watching this video, I am quite sure, hopefully, that you learned, studied about the sodium-potassium pump. Let's draw a few of those in our picture to keep us honest, okay? I'll use light green. I'll put it right here as a big old globular protein. Sodium, potassium, pump. That active transport pump that pumps sodiums out of my cell, potassiums into my cell. And I'll put a few on here like so. Here's one. I'll just scatter them around wherever, makes you happy. Some sodium, potassium. So you understand that they are here. Even one in the T-Tubule there. Some sodium, potassium pumps. And what do? Pump sodium and potassium. 3 sodiums out. And what? 2 potassiums in. I'll just give myself a little picture down here. 3 sodiums out and 2 potassiums in. This is the sodium-potassium pump. Setting up a situation where I have a whole gob of sodium outside the cell, none inside, a whole bunch of potassium inside the cell, hardly any outside. So let's draw in some sodiums and potassiums, everybody. And, oh yes, I'm making a big mess out of this picture. That's perfectly fine, because guess what? It's my picture. I can do that. So outside my cell, I'm going to draw in some sodium ions. Na plus 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, and I'm going to put a circle around each one just so they're easier to see. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Now I always do this. I write them out and then I draw circles around them so I know I get the number correct. So there's 12 of them. What number is 2/3 of 12? Well, that would be 8. So that's how many potassiums I'll draw inside my cell. 1, 2, 3, 4, 5, 6, 7, 8, 8 potassiums inside. Yeah, it's not as pretty anymore, is it? That's all right. 8 potassiums inside as compared to 12 sodiums outside. I have established a resting membrane potential in this cell of what? -85, -90 millivolts. So the resting membrane potential, thank you sodium potassium pump, in a skeletal muscle cell about -85 or -90 millivolts is the way we describe it. And I'm going to ask the question that I ask all the time repeatedly about this picture we're looking at. Why? Why does the sodium potassium pump out all these sodiums? You know the answer. Sounds silly, I know, but I think you know the answer. Why does the sodium potassium pump spend ATPs and all this time pumping all these sodiums out of the cell? So we can let them come back in. That's why we do it. That is literally why we do it. We pump all these sodiums out of our cell only to turn around and allow them to come back in. Think about it. So this motor neuron, part of our nervous system, when we decide to, we will spit out acetylcholine onto these ligand-gated sodium ion channels that you see here. So here comes the motor neuron. It spits out acetylcholine like that. Acetylcholine, 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 making these ligand-gated sodium ion channels open. Once they're open, sodium will rush through these open doorways. It's not just going to meander, it's not going to ooze in there because we've set up this large concentration gradient with many sodiums outside, few inside, plus an electrical gradient, more positively charged particles outside than we have inside. So these sodiums are going to come sweeping in through these open doors very, very quickly, aren't they? Whoosh, 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 like that. Which creates a big electrical event that happens right here at what's called the neuromuscular junction. This big influx of sodiums, whoosh, right here, whoosh, through these open doorways, creates what we call a local or a graded potential. This is a local or a graded potential. Sodium ions rushing in, whoosh, right here. I will even label it for you on my picture. I don't think you have to in such a big mess, but this is where we have the local or the graded potential, which is right here with the opening of these ligand-gated sodium ion channels and sodium, whoosh, you need a sound effect, whooshing in. If enough sodium comes in right here to change this resting membrane potential, positive ions coming in, we're getting more positive, to somewhere up around the negative 55 millivolt range. You'll study this in your lectures. That's threshold. That, everyone, is enough to trigger the opening of this first voltage-gated sodium ion channel. See that one? It's blue, voltage-gated sodium. Remember those blue. If enough sodium comes in during this local or graded potential, that will trigger the opening of the first voltage-gated sodium ion channel. Now, I'm going to scroll down from my picture just a little bit to get a little blank space. I'm going to come back to it because it's so nice I can't help but look at it. But I want you to think of a set of dominoes. I'll just make 3. Domino 1, domino 2, domino 3. If I want to make domino 1 fall, I do that with my finger, don't I? You know what, everybody? I can't draw lots of things well, but I can draw a finger. Look at that. Perfectly good finger, right? So if I knock over domino number 1, then it's going to knock over domino number 2, which is going to knock over domino number 3, and then domino number 3 is going to fall. Like that. But I never touched domino 3, did I? I only touched domino number 1. So this falling of domino number 1, this is like my local or graded potential. Does enough sodium come in to make the first voltage-gated sodium ion channel open? Did I get enough of a change to make number 1 open? Because if number 1 opens, then number 2 opens, then number 3 opens, and so on. Once the dominoes start to fall, that's what we call an action potential in this cell. So back up to my cell. Oh man, is this a mess, isn't it? I love it. This is good. This is what we like when we're studying. So if the first voltage-gated sodium ion channel opens, then wouldn't more sodium come in through that one? Here, I'll use a standout color like rose gold. I kind of like that. So wouldn't more sodium now come in through this voltage-gated channel? And then if the first one opens, so does the second one, so does the third one, so does the fourth one, and the 778th one. So do you see what happens here? I have created this falling set of dominoes where every voltage-gated sodium ion channel is going to open, and sodium's going to be whooshing in all over the sarcolemma and whoosh down the T-tubules. Got to have that whooshing. This is called an action potential. This in rose gold is an action potential that we always describe using terms like this: a self-propagating, unstoppable wave that's going to run all over the sarcolemma and whoosh down the T-tubules. This is the voltage wave I was after, everyone. This is the voltage wave I was after. So if I spit out acetylcholine, opening the ligand-gated sodium ion channels, sodium rushes in here. You don't have to draw this and make this big mess, but sodium rushes in here at the neuromuscular junction. I'm just going to pick a color that might stand out a little more. If I can, I'm going to have to go with my super colors. How about bronze? So if I spit out acetylcholine, I get that local potential going on right here. And if that's big enough, then I get the action potential happening all over the sarcolemma and down the T-tubules. And that action potential whooshing down the T-tubules is what will trigger the release of these calcium ions. Now, I said I was going to draw this in a little more detail for us. I would like you to look very carefully, everybody, at the diagrams you have in your textbook, because here you see some variation in books depending on what your textbook chooses to show you. So I'm going to draw my T-tubule much larger here. This is my T-tubule. I'll use the same colors we used before. Here's my sarcoplasmic reticulum right there, like this, with all my calciums in there. And I would have voltage-gated sodium ion channels right here, right here, right here. Now, I want you to understand the physiology going on here. People are not quite sure, meaning science people, not quite sure how far down do these voltage-gated sodium ion channels go. It's different in different species. It's different in skeletal versus cardiac muscle. There's some argument people have here. But what we don't really argue about is what occurs here. So if I go back to my super cool lava color, there it is. This represented my voltage-gated calcium channels, didn't it? And these things are linked to the T-tubule membrane right here. They're linked to it. Even more close up. Here's the T-tubule membrane. Lava. Here's a voltage-gated calcium ion channel. And I'll even use a different color here. Ocean. Well, that's kind of cool. Ocean. And then there's a linking protein right here. This is a linking protein, and it is voltage sensitive. So when that voltage wave, remember we called it the action potential, when that voltage wave came sweeping down the T-tubules like that, it actually mechanically pulls on this protein link, causing this thing to open, and now calcium can get out. So we can argue all day long, muscle nerds like myself, about how far down do the voltage-gated sodium ion channels go, but it's not really that significant for us in terms of what happens. So these voltage-gated calcium ion channels of the sarcoplasmic reticulum, because they'd be part of the sarcoplasmic reticulum's membrane like this. This is what I'm drawing over here. These protein links respond to the voltage wave and pull this door open, this voltage-gated calcium ion channel, allowing the calcium to leave. This is what you see in your textbooks called the excitation, contraction, coupling idea. This change of an electrochemical event, a voltage wave, sweeping down the T-tubules, everybody. Here's that voltage wave down the T-tubule, changing that into the actual opening of a door, a voltage-gated calcium ion channel. And then what happens? The calcium is allowed out. The calcium binds to troponin. The troponins pivot, roll over, fall down, and the tropomyosins are pulled off of the active sites. What I'd like to do before I leave my mess of a picture is go through some of these steps with you one time. You might have a set of steps written somewhere that describe each of these actions. So follow along looking at my picture. A motor neuron in the upper left releases acetylcholine into the synaptic cleft, the little gap. I can erase, I hope, without making too big a mess. A motor neuron releases acetylcholine into the synaptic cleft. Sorry about that. I got to get my eraser going. There we go. Into the synaptic cleft of the neuromuscular junction. The acetylcholine binds to ligand-gated sodium ion channels found there on the sarcolemma, causing them to open. Sodium that was previously pumped out by the sodium-potassium pump, thank you sodium-potassium pump, will rush into the cell, creating a local or a graded potential in that area. Local or a graded potential right here, everyone. If the local potential is big enough, meaning if I change the membrane potential from about negative 85 or 90 up to negative 55, that will be enough to trigger the opening of the first voltage-gated sodium ion channel. And once the first voltage-gated sodium ion channel opens, they will all open in a self-propagating, unstoppable wave all across the sarcolemma and down the T-tubules. This is called an action potential. This voltage wave flying down the T-tubule will trigger the opening of the voltage-gated calcium ion channels of the sarcoplasmic reticulum, allowing calcium that was previously stored here to leave, enter the sarcoplasm, and bind to the troponin proteins, causing the troponins to fall over, pivot, rotate, fall down, whatever you like to say, which pulls, heaves those tropomyosin ropes off of the active sites. Once the active sites on the actin proteins are exposed, then the myosin heads are allowed to grab them and pull them toward the center of the sarcomere, taking up those empty spaces, those sliding filaments, making each and every sarcomere shorter all at once. And when these sarcomeres shorten, they shorten 100% all the way as much as they possibly can. The way this works is that muscle cells, skeletal muscle cells, contract all the way. That means every sarcomere every myofibril, the cell gets as short as it can possibly get, or not at all. In your lectures, you will call this the all or nothing principle, true for muscle fibers and neurons. They either go all the way 100% or they do nothing at all. So the entire skeletal muscle cell contracts as much as it possibly can, or it doesn't do anything at all. Those are the only two choices we get. Hopefully, watching me make this mess of a diagram is a way for you to study the parts and then the physiology of these skeletal muscle cells. Now remember, in closing, about the picture, I told you this is a highly stylized picture of a skeletal muscle fiber. I suggest you draw it multiple times or watch this particular video segment multiple times, and it is my hope that it helps you understand what's going on here. But for now, all you A&P nerds, let's continue on in our own lectures and learn more about this stuff, because I know you really want to.